Hello, my name is Rachel Tallis. I'm a field application scientist supporting Bio-Techne single cell dispensers. Thank you for joining me today for a virtual demonstration of Pala, our easy-to-use, ultra-gentle cell dispenser. The Pala simplifies many single-cell applications, including cell line development, single-cell genomics, CRISPR editing, antibody discovery, and rare cell isolation. Our proprietary microfluidic technology combines flow cytometry and liquid dispensing to sort single cells directly into 96 or 384 well plates. This technology allows for efficient single-cell sorting that is 20 times gentler than a traditional fax machine. Operation is simple and fast. The system provides an intuitive interface that guides users through every step of the process, taking you from startup to dispensing in less than 10 minutes. Your cells are loaded into sterile, single-use cartridges, which keeps your samples sterile and eliminates sample-to-sample carryover. All of this in a box about the size of a desktop printer. The Pala is portable and can easily be moved into or out of a tissue culture hood as needed. The Pala is equipped with 2 excitation lasers that can be configured in either a 488/405 option or as a 488/561 option. That allows for 2 light scatter measurements and between 9 to 11 fluorescent channel measurements. Today, we'll be dispensing cells with the 488/561 Pala. We will be dispensing CHO cells, a common cell line used in biologics manufacturing. Our samples are a mixture of wild-type and green fluorescent protein-expressing cells. We will also stain these cells with Cell Tracker Red to identify live cells in the population. We recommend using a viability dye to improve experimental success in single-cell outgrowth. When we set up the Pala, we will use light scatter and green and red fluorescence to sort the cells of interest into a 96-well plate prepared with media. After sorting, we will image the plate to confirm the efficacy of our sort. First thing I'm going to do is turn on the instrument and open the lid so we can take a tour of the system. This is the stage. This is where we can place a micro-titer plate for single-cell dispensing, or we can use the bulk adapter to hold a microcentrifuge tube for bulk cell sorting. This is the cartridge holder. It will open and close automatically during use of the system through a series of software prompts. If the lever is up, then the holder is open. If the lever is pointed to the side, it is closed. We also provide a cleaning cartridge. The cleaning cartridge enables the instrument to clean its fluidic lines during certain steps of normal operation. Now, let's take a look at the software. The software interface uses icons and prompts to guide you through the process. To start my day, I am going to navigate to the upper right-hand corner and click on the Start button. This will begin the initialization of the system. Here, we will encounter our first software prompt. I will not click on OK until I have completed the action described in the prompt. This prompt has instructed me to load sheath fluid into the sheath bottle. The sheath and waste bottles are in the bottle holder on the side of the instrument. The bottle holder is equipped with a pair of scales, and the software will alert you if either bottle is empty or full. For single-cell sorting, we recommend using sterile filtered water or PBS, but serum-free media can also be used. Next, place the cleaning cartridge in the cartridge holder. After clicking OK, we will see the cartridge holder close and the instrument will begin its initialization procedure, which takes about 5 minutes. In the bottom left-hand corner of the software, we can see that the status of the instrument has changed and a progress bar has started. We will also be able to see the waistline drip as sheath fluid begins to flow through the instrument. While the instrument is starting up, we can get our cells ready and look at the single-cell cartridge. The cartridges are single-use, but can be reloaded with more of the same cell sample as needed. Each cartridge holds up to 600 microliters. For this experiment, I have diluted my CHO cells to a concentration of 10,000 cells per milliliter, and I will load 400 microliters to the top port to fill the reservoir. During a cell sort, the pressure in the cartridge is tightly regulated and never exceeds 2 psi as cells move through the flow cell. Compare that to other traditional cell sorters that can go as high as 35 psi. Now that the instrument has finished initialization, we will be prompted to select either single or bulk sorting. I am going to select single cell. The cartridge holder opens and it is time to insert our cartridge. Notice that I'm only touching the sides of the cartridge, avoiding the front where sample will be interrogated by the lasers. The cartridge must be inserted in the proper orientation, matching the label on the cartridge holder. I give it a quick tap to ensure the cartridge is fully seated, and we're ready to go. The software will now prompt me to put the waste collector on the stage. When I hit OK, the instrument will prime the cartridge and start the flow of sample through the microfluidic channel. At this point, a live camera image will appear that will allow us to properly align the microfluidic channel to the laser. Using this silver knob, I will adjust the image until the left white line is aligned with the side wall of the dispensing channel. Once this is set, I will click on OK to proceed. Next, we will do a quick inspection to ensure that the nozzle on the cartridge is dry. A droplet on the nozzle indicates that the cartridge needs to be reprimed. Prior to sorting, we will first analyze our cells. In order to visualize our cell populations, we will need to set the appropriate charts. The first chart is always set by default and displays forward scatter by total scatter, or FSC versus TSC. That will give us a look at the size and complexity of the cells. We can then focus on analyzing GFP. We will select a scatter plot using the FITC channel and forward scatter. The third chart will be for our cell tracker red, and we will be using the PE channel for this dye. For this channel, we will plot the events using the histogram chart type and plot PE by itself. Now that I've set my charts, I am ready to analyze my cells. I am going to go up to the menu bar and click the Analyze icon. The Pala will begin analyzing the cells and the charts will populate with events. Down here, in the lower left-hand corner, the software is reporting the event rate, time, and total events for this sort. While the instrument is analyzing cells, I'm going to ensure that the event rate is between 1 to 3 events per second. That will give me the best purity during my dispense. Recall that I had previously added cells to the cartridge at a concentration of 10,000 cells per milliliter. To change the event rate, I can adjust the sample flow rate using the drop-down here. On the FSC versus TSC plot, we can see that the cells form a nice cluster. If we right-click that cluster, we will get a list of options for gating. I'm going to select Create Gate. Once a gate is created, it can be edited in a number of ways. It can be resized, moved, or vertices can be added or adjusted. Next, let's gate for the cells that express GFP. Over here, on the FSC versus FITC plot, we can see a range of intensity of GFP within the CHO cell population. We will add a gate and adjust it so that we will collect the cells with the brightest fluorescence. For cell tracker red, we will create a gate in the PE channel to capture the cells with the brightest red fluorescence. That will be our live cell population. The location of the gates can be adjusted by clicking and dragging. The gates are additive, which means that only the cells that are inside of the first, second, and third gate will be dispensed. Any cells that fall outside of these 3 gates will not be dispensed. Now, let's define where we will deposit our cells. The software has options for 96 or 384 well plates, and wells for dispensing can be selected individually, in groups, or all at once. For this experiment, I'm going to select all. Now that we're ready to sort, I'm going to get my plate out of the incubator. I have prepared my 96 well microtiter plate by filling each well with 150 microliters of media. At this point, I will stop analysis by clicking again on the analysis button. To start the single cell dispense, I'm going to click on the single droplet icon. The software will prompt me to load the microtiter plate onto the stage, ensuring that the A1 position is in the upper left-hand corner, and then I will hit OK. As cells travel down the microfluidic channel, they will be interrogated by the excitation lasers. Cells that meet the fluorescent and scatter criteria defined in the software will activate a high-speed valve that will push the cell into the dispensing channel. The cell is dispensed in a 1 microliter droplet into the well with media, and a corresponding checkmark will appear in the software to tell us that a cell has been deposited into the well. Dispensing time will vary depending on the abundance of your target cell, but it typically takes about 2 minutes to fully dispense a 96 well plate, or about 6 minutes for a 384 well plate. Generally, we can observe outgrowth a week after sorting. Here's an example of colony outgrowth from cells dispensed by the Pala. Now, I am going to switch to a new sample. These are also CHO cells, but they have a lower proportion of GFP-positive cells. I'm going to hit the switch button on the menu bar. The software will prompt me to put in the cleaning cartridge. When I hit OK, the system will run sheath through to prevent any sample-to-sample carryover. Switching cartridges takes approximately 2 minutes, which gives me time to fill my next cartridge. I'm also adding 400 microliters of cells at 10,000 cells per milliliter. We will prime this cartridge, align the channel, and check the nozzle again. Since we are using the same charts, we are ready to hit analyze. As the charts populate, we can appreciate the differences between the two samples. We're not seeing as many highly fluorescent events due to the lower abundance of GFP-positive cells, which is what we expected. We were able to switch to a new sample in just a few minutes without concern for cross-contamination or sample-to-sample carryover. If I wanted to dispense these cells, I could use the same gates or draw new ones, depending on the needs of my experiment. When we're done with Pala for the day, I can start the shutdown procedure by selecting the red stop button in the upper right-hand corner of the software. The software will ask me to fill the sheath bottle with sterile filtered water. The sheath bottle is already full of sterile water, but if I was using PBS to sort, I would swap the sheath fluid now. The instrument has completed the shutdown cycle. We can close the software, turn off the unit, and dispose of the fluid in the waste bottle. Thank you for joining me today for our virtual demonstration of the Pala benchtop cell sorter. I hope I was able to demonstrate how simple, flexible, and powerful the Pala is. If you have any questions about getting a Pala for your lab, or if you want to learn more, please find us at bio-techne.com/Pala. You will be able to find the specs of our instrument, customer reviews, and how to contact your local representative. Thank you again, and I hope you have a great day.